Good morning, everybody.
Good morning.
Good morning. So, last lecture we have started with the plant propagation. So, the plant propagation is possible through by using the seeds as well as by using the vegetative plant parts. So, the propagation is multiplication of plants by using plant propagules. The propagules means the plant part which is having the ability to develop the new plant. So that may be possible through the seeds or by using the vegetative parts. So under the vegetative parts, there are several methods available to do the propagation, cutting, grafting, budding, layering, specialized organ, and for the mass propagation, we can go for the tissue culture. Then we started with the sexual propagation. The sexual propagation is the multiplication of plants by using the seed. So seed propagation or sexual propagation like that we can call. Then if you look into the advantage, because some advantages we are having by going for the sexual propagation also, because that is simple method. Even we can produce large amount of seedlings within the short period and short space. The storage of the material easy. It is having the even long live, that type of advantages we are having. But even disadvantages also there, because these plants are spreading type and management practices for these plants difficult. Even we can't produce the true type because there is the variability due to the cross pollination. But for the breeding purpose, so this is better because we can incorporate some superior characters and get it through the hybridization program. Then even in sexual propagation, because the management practices difficult, even juvenile period of the plant is long, so that type of disadvantages we are having. But in advantages, the plants are able to even disadvantage some plants we can't produce the seeds, so that type of plants it is not suitable method to go for the propagation. So advantages as well as disadvantages yeah in the sexual propagation. Then in asexual propagation, so this is the method even we can adapt some plants which is not producing the seeds. Even we can produce the plant prototype. The management practices easy. The juvenile period or the vegetative period is less, so we can get the products quickly compared to the seed. So that type of advantages we are having, but some disadvantages there, because in grafting and budding the technical skill needed, even the maintenance of the nursery also needed, so that type of disadvantages also there. Then we move to the cuttings. So that is one of the method we utilized in the vegetative propagation. Cutting, we can take the cuttings from any plant part, that may be the leaf or stem or root. So from which plant part we are taking the cutting, that name is we call. So stem cutting, root cutting, but leaf cutting, leaf bud cutting. Then if you look into the stem cutting, from which portion of the stem we are taking, whether it is the mature portion or the semi-mature portion or the before the lignification, if we check, so that also further classified into four: hardwood, semi-hardwood, softwood, and herbaceous cutting. Hardwood cuttings means we are taking the cuttings from the mature wood. So that is especially in the deciduous plants, they are having the 4 seasons. So during the late winter, we can take the cuttings from that type of plants, that is hardwood. Even semi-hardwood means these cuttings can be taken from partially mature wood. Then softwood means this is also, we can take the cuttings from the wood, but before the lignification we can get. That is called as softwood cutting. Then herbaceous means this type of cutting can be taken from succulent plant. So I put some examples also. That is the stem cutting. Then in leaf cutting, so if we make the cut on the midrib of the leaf, and if we plant it in the growing media, that leaf portion is in a position to develop as the new plant. So African violet, begonia, peperonia, bryophyllum, this type of plants can be propagated through the leaf cutting. Even for the stem cutting, crotons, show flower, jasmine, coleus, grapes, rose. So depending on that, which plant portion we are taking the cutting, stem portion we are taking the cutting, so that can further classified and example be studied. But in leaf cutting, so if we make the cut on the midrib of the leaf and a small portion of the leaf with the mid rib, if we plant it on the media, we able to produce. Then root cutting, so from the root, so it can spread under the soil, and it can give the new plants like curry leaves, breadfruit, jackfruit. So that type of plants also giving the new plants through the roots. Then another one leaf bud cutting. Here we are taking the cuttings with the small portion of the uh, leaf as well as the bud, a small portion of the stem, leaf and bud. So leaf bud cutting. This is commonly practiced in lemon tea. So that type of plants we are practicing. Excuse me. Sorry, additional director Arasa Kesari sir took the call. That's why I answered. Um, because in cuttings, so if we can propagate through the rooting also, root also, then leaf buds. So there are several methods available in cutting. So in cuttings, next I think we studied about the roots, the two type of roots, the pre-formed roots and wound-induced roots. Preformed root means that roots are already available in the dormant stage. So if we make the cutting, so that is in a position to develop. Wound induced cutting means after making the cut, the callus is formed in that cut portion, and then root initials develop from the callus. Callus is the mass of parenchymatous cells, irregular mass of parenchymatous cells. We already studied that. So from that, so that two types of roots we able to see in the cutting. Then the factors which influence or affecting the cuttings, there are several factors, even condition of the bud, the time of season. There are several factors affecting the cutting. So we studied that last class. nearly 17 points we have studied under the factors. I will put, after finishing these lectures, I will put the lecture notes on the LMS for the first and second lecture. So today we will continue with the grafting. So the grafting is the technique we are using to join the two plant parts. So the top plant parts we are calling that as the scion and the bottom plant part as the rootstock. So we are connecting the cambium of the scion as well as rootstock and make them to grow as the single individual plant. So that is the grafting. We are using the techniques to join the two plants cambium. So that plant's rootstock and scion. So rootstock means the plant part which is giving the bottom of the plant, and scion means the plant part which is taken from the another plant and unite together in as the single plant. So this is giving the top portion of the plant. So here in grafting, what we are making the wound, and we try to join scion into the rootstock by joining the cambium and make them to grow as the single plant. That is the grafting. Here we have to know about the scion, the short piece of a plant part that is separated from the desirable character mother plant, utilized as the terminal. Because after doing the grafting, it is giving the top portion, terminal portion. The rootstock is the lower portion of the graft. So it is having the root system of the grafted plant. Why we are having the rootstock? Because while selecting the plants for rootstock, that is having some ability, good rooting ability, tolerant to some environmental condition, even tolerant to some pests and diseases. So that type of plant, even we can take it as the plant for the rootstock. Scion is the top portion that should have the more desirable character because that is the part growing and giving the productivity. Then intermediate stock, that term also we have to know. In some cases, while doing the grafting or budding, if we put the scion into the rootstock, that scion won't grow successfully. So that case, another small piece of stem is needed to combine the rootstock as well as the scion. So that stem part is called as intermediate stock. Then we have to know about the vascular cambium because the thin tissue that is located between bark and the wood is the cambium. So these cells, normally meristematic cells, they are in a position to dividing and form the new cells. So if we make the wound by cutting the cambium, so it is developed as it's dividing and forming the new cells. Due to that, in grafting and budding, we are making the wound and combining the two cambiums of the rootstock and scion. So they undergoes to division and forming the new cell and make the wound heal or that is developing as the new plant. So for the successful union, the cambium of the scion that should be placed in close contact with the cambium of the rootstock. So while doing the grafting or budding, what we have to do? We have to make the close contact of the rootstock and cambium. Then only it is in a position to divide and heal the wound. So then it is developed as the single plant. The callus, irregular mass of parenchymatous said. So normally that is in cuttings we studied. So it is developed from the wound plant. So it is in the position to make the wound healing. So that is the case. So if we look into the stages in grafting, first what we have to do, we have to take the grow the rootstock. So after growing rootstock, what we have to do, we have to get ready of the scion which is having the superior character. So here we have to remove the leaves on the scion and we have to get the scion ready. Here then we can make the cut on the rootstock. Then insert the scion into the rootstock by joining the cambium of the scion as well as the rootstock. So after inserting, so we have to wrap this to avoid the water going inside, and we tightly wrap this by using the polythene. So after 2 weeks, if the successful union or the callus formed and successful union happen, so this shoot is in a position to grow as the new. So that is the situation. So if you look into the advantages of cutting, so this is the better method, grafting, sorry, advantages of grafting. This is the better method because we can develop the plants that is with the good characters. Normally we know the vegetatively propagated plants are very quick in bearing. Best rootstock we can utilize. Number of variety we already know that can be grafted in one plant. Damage happen to plants also can be repaired by going for the bridge grafting. Even the resistance to disease, that character can be developed from the rootstock. Even the precocity, that is the ability to induce fruitfulness without the need for the completing the unit. That means while vegetative growth happening, we can get the some reproductive, promote the reproductive growth, and we can get some product rather than the completing the vegetative growth or the juvenile growth. Be able to induce the fruiting. So that is also possible in grafting. Shape of the plant that can be changed because if the plant is larger one, we can do the grafting by cutting and insert the scion by using the dwarf varieties. That is possible. So this for the this type propagation, the skill is needed, but we can do that easily. We can maintain the genetic consistency because fruit type plants we can develop. So that is because in apples, some genetic variability even we can observe. So that case, if we go for the grafting, so we can obtain the maintain the genetic purity. Then change of cultivars also possible because if the plants become older, if we go for the top working, so even by inserting the new scion in the higher canopy plant, we can get that. So that is possible. So changing of cultivars also possible. These are some advantages in the grafting. Then if you look into the disadvantages, here we can't do the grafting or budding for the old plants. So because if we want to obtain the successful grafting or budding, the same family plants can be combined. So the compatible nature should be there. So we can't utilize this method for the older plants. This is more successful, especially in dicot plants. New variety we can't develop. We can develop the plants which is similar to the mother plant, because from which we are taking the sign. So this is the extensive, that is a broad method. But for this method, to join the KMPM of the rootstock and scion, that skill is needed. So a specialized skill manpower is required compared to the cuttings. The lifespan of the grafted or vegetatively propagated plant is short compared to the seed propagated plant. So yeah, but in here we are having the advantage, some resistanceness to the diseases also possible, but some susceptible virus diseases also can be spread. So that is also the problem. Even tolerant to environmental conditions, so that is we can obtain through the rootstock. Anyhow, some problems also there. Then we will see, because we are talking about grafting, so then rootstock, science. So what are the advantages and disadvantages we are having in grafting? So we have to know about what is graft union. The successful formation of the union is the basic requirement for the successful grafting. Formation of graft union means if we combine the rootstock and the scion cambium together and tie up with the polythene, so that wound made, that can be healed and develop as the single plant. So that combining of the rootstock and scion is by doing the grafting and successfulness. So that portion is called as graft union. The grafting the rootstock and scion that are placed in close contact with rootstock as well as in scion, and we allow them to grow as the single plants by tidying up or the wrapping up with the polythene. So the formation of a graft union, that is the basic process of healing of wounds, because we are making the injury to rootstock and injury to the scion and inserting inside the rootstock, we are inserting the scion. So the two wound portions, cambium, should be joined together. If the union is success, then it is in a position to develop as the single plant. So if the healing is completed, so there is a chance for the translocation of assimilates from the roots to shoot. So re-establishment of the two plant parts, that is possible. So if we want to obtain the successful grafting, what are the basic, the general requirement we will look into. So the first requirement to obtain the successful grafting is compatibility. That means if we join the rootstock and the scion, that should have the ability to join as the single plant. So here, if we make that, we have to take the cuttings from the scion and the rootstock, that should be from the same family. then only the characters can match. So the matching of tissue. That means here, if you want to obtain the successful grafting, the cambium of the rootstock and the sign should be matched properly, close contact. Then only it can be developed as the single. So compatibility should be there. That is the same family crop we have to select as the rootstock and the scion. At the same time, matching of tissue should be there. In addition to that, the desiccation of the grafted portion should be avoided. Desiccation means removal of moisture from the grafted portion. So immediately after grafting, the cut surface must be covered with the waterproof material. So that is the case. We are wrapping with the polythene on the wrapping strip of the polythene. Or we can put some bags or the plastic tights or the rubber tights we can utilize. Here we have to avoid the moisture loss, desiccation, moisture loss from the grafted portion. Then season is very important. So the grafting, that operation should be done at the right time of the season. That is also very important to obtain the successful union. The rootstock and scion that should be in the proper stage of the maturity and the growth. Then only they are able to grow as the single plant. Otherwise, it won't grow. So season is very important. So the right time we have to select the rootstock as well as scion, and with the proper stage and the maturity we have to do the grafting. Then pressure, because this is also very important. Right stage of season means because while taking the scion as well as making the cut on the rootstock, that time the cambium cells should be in active division state. So that is the best time because easily the wound can be healed. So that is also we have to consider season. Then pressure, because sufficient pressure between the rootstock and scion that is required to prevent them from moving independently of each other. Because if the pressure is in a suitable level, so there is a chance of healing of the wound and the translocation is possible quickly from the rootstock to the scion. The other factors also being generally required, the weather condition, the pests and diseases, because while taking the scion, we have to select the scion from superior mother plant, and that mother plant should be free from pest and disease attack. So that is very important. Even while offshoots appearing below the graft union, that means if we make the cut on the rootstock and insert the scion and do the grafting. Then the rootstock portion is started to develop. What we have to do, so to promote the shoot growth, we have to clip off or the remove the shoots that is developed from the rootstock. So only the grafted top portion we have to promote the growth. So that type of requirement also there. Then in grafting, so in grafting, what is grafting? What is scion? What is rootstock? What is intermediate stock? Then what are the advantages and disadvantages we are having by going for the grafting? Then what are the general, how we are forming the graft union, and what are the general requirements we need to obtain the successful grafting. So basically the same family crop, matching of cambium, rootstock and cyan. Then we have to think about the season when we are taking the cutting, the cyan and making the cut on the rootstock. So that time the cyan should be in active dividing condition. So that should consist the active parts also. So that type of things we have to consider. In addition to that, the pressure flow between the rootstock and cyan, that is also influencing the healing. Then, while promoting the shoot growth on the cyan, so if any shoot growth that is start after grafting from the rootstock, that should be removed. These are the things mainly we have to consider. Then we will see what are the methods of grafting possible. Mainly, two methods available, attached method and the detached method. Attached method means while the in rootstock, we are doing the grafting, not separating as the stem portion. So the two plants we combine together by making the wound. We are combining together and allow them to grow as the single. After successful union and growth, so which part we want to take it as the top portion, we allow from the one plant. In other plant, if we want to develop that as the rootstock, leave that. Then other portions we have to cut off. So that is the case. Here in attached method, the two plants, they are having their own root system. So here also we are having some types in hatching method, approach method, bridge grafting, that is attached method of grafting. Then in detached method, whip grafting, tongue grafting, side grafting, notch grafting, wedge grafting, bark grafting, saddle grafting. Because shape of the cut we are making on the rootstock and cyan, these names are given. And here we are separating the cyan from the mother plant. But in attached method, we are not separating the cyan top portion from the mother plant. We are using the two plants, both are having the own root system. After successful union only, we are cut off in the plant part which is needed to us. So then the next thing I will show the pictures how these things we can have. Then we will see graft incompatibility. That means if we do the grafting, the rootstock and cyan, they are not in a position to develop as the new plant. So that nature we are calling that as incompatibility, that is inability or the failure of two different plant parts, that is rootstock and cyan, joined together to develop as the new plant by forming the successful union. That was not happen if there is incompatibility nature. That is, if we join that, if the grafted portion not grow properly, successfully, that nature is called as incompatibility. Because if you want to grow as success, that should be compatible. That is same family like that. So incompatible is the opposite of the compatible. So it is unable to form the successful union. So how can we identify? Because this is due to the incompatibility. So the symptoms we able to see on the grafted plant. So what are the symptoms we can see? Complete failure to form the bud union or the graft union. Because if it join together rootstock and scion, it won't grow. So that is one symptom due to the incompatibility. In some cases, they can form the union and initially they grow, but later stage it can die permanently. So initially it shows as the success, but it won't continue the growth. So that is due to the incompatibility. In another case, even vegetative growth and premature defoliation, that is, young leaves started to fall off, and vegetative growth also decline. So that is also symptom of incompatibility. In addition to that symptoms, some differences in rootstock and scion be able to see in size, appearance of deficiency symptom, even in stunted growth, even yellowing of foliage. So the in cut portion or joint portion over or below the swelling of that, because the translocation is affected. Due to that, this is showing this nature. Initially, it shows it is success, but later stage, because the grafted portion is showing the swelling because of the translocation effect. So these are some symptoms we can see on the incompatibility if happens in the graft or budded portion. Then the types of incompatibility also there. So localized incompatibility, translocated incompatibility, virus-induced incompatibility. There are three types of incompatibility possible in grafted portion: localized incompatibility, translocated incompatibility, and virus-induced incompatibility. So the localized incompatibility is due to the physiological processes influence or affected. Translocated incompatibility, especially the degradation of the phloem happen due to the degradation of the phloem. So this is happening. Then virus-induced incompatibility due to that, the presence of virus in the plant. So first we will look into the localized incompatibility. So this type of incompatibility, so reactions that is depend upon the actual contact between the rootstock and side. So if the contact is not happen properly, so it is caused the localized incompatibility. Because if we want to obtain the successful graft union and single plant, we know the kpm of the rootstock and the scion should be combined properly. Excuse me, Nalinanis is taking. Sorry. So the three types of incompatibility possible: localized incompatibility, translocated incompatibility, and the virus-induced incompatibility. So under that localized incompatibility, if you look into uh, So Dean is taking calls for it. From the Dean. So if you look into the types of incompatibility, there are three types I already mentioned: localized, translocated, and virus induced. Localized incompatibility means here the joining of the two cambiums of the rootstock and cyan is having the problem. So that case, I already told you, if there is a problem of joining of rootstock and cyan, we can use the intermediate stock. Another stem portion we can use in between the rootstock and cyan. So this type of localized incompatible problem can be overcome by using the intermediate stock. Then translocated incompatibility. So if the union is a form success, then KMPM is joined properly, then the rootstock, phloem of the rootstock and the phloem of the cyan join. So that translocation will happen. So some degradation happen in the phloem. This is the part because normally we know in the plants we are having the xylem and phloem. Xylem is responsible for the absorption of water and nutrients from the rootstock and translocating. And phloem is responsible to translocate the assimilase, the product that is produced through the photosynthesis from the leaf. So we know the two vessels where the plants are having phloem and xylem. So the phloem is important for the translocation of assimilase. So some degradation or deterioration happen in the phloem. So that will influence the translocation because translocation won't happen. So that incompatibility can be called as translocated incompatibility. Then the presence of some viruses also possible through the rootstock to the cyan, I already told you. So if that happens due to the presence of virus, so the incompatibility can be promoted by the virus. So that case, it is called as virus-induced incompatibility. So these are the types of incompatibility: localized incompatibility, translocated incompatibility, and virus-induced incompatibility. Then we will see the causes of graft incompatibility. What are the causes may be possible? So that is due to the anatomical structure or physiological and biochemical reasons available in the plant, or nutritional deficiency, even presence of virus that can cause the incompatibility. First, we see what are the anatomical and structural reasons that can influence the incompatibility. So the rootstock and science may not be differed structurally, but abnormality that may be present due to the some parenchymatous cells at the graft union. So it can affect the continuity of the vascular translocation. So that can influence the incompatibility. So that case, the structural changes happen in the grafted union that is causing the incompatibility. So that is the thing we have to study. So due to this structural change, the discontinuity, the phloem won't translocate the assimilates to the rootstock and other parts. So that is possible. Then physiological and biochemical reasons. So here the failure of the phloem or xylem at the graft union. So that is causing the starvation of the root or the wilting of the scion. So the some problems occur between the vascular cambium of the rootstock and scion. So the physiological activities like photosynthesis, assimilate can't be translocated, and also then water and nutrient can't be translocated. Then some nutrition deficiency, if we observe, that also can cause the problem. Then presence of viruses, that is also causing the problem of the incompatibility, like mycoplasma-like pathogens present, latent virus or the mycoplasma-like pathogens present, that may influence the success of the graft union. So that is regarding the incompatibility. Incompatibility is the inability to form the successful union. So in incompatibility happen, some symptoms we able to see in the grafted portion also. Then what are the types of incompatibility possible? Localized, translocated, and virus induced. For that type of incompatibility, what are the causes? Some physiological causes may be there, some anatomical structural change because parenchymatous cells irregularly formed, that is preventing the translocation, or some physiological or biochemical reasons, or translocation of water and food will be affected, or some presence of viruses, or some nutritional deficiency also can cause. So these are the reasons for causing the incompatibility. Then we will see, similar to the factors influencing the cutting, here also what are the factors influencing the grafting? We have to look into plant species and type of graft, environmental conditions like temperature, moisture, oxygen supply is influencing, growth and activity of rootstock. Then craftsmanship, that means technical skill person and grafting ability. Then polarity, because if we do the grafting in stem portion above ground part, we have to think about the polarity. And even if we do the grafting in root part, we have to think in other way regarding the polarity. So we will study detail in other slides. Then virus contamination also influence. Then incompatibility that can causing. Then plant growth regulators and the graft union formation can influence. That other factors influence the grafting. So under the first factor, plant species and type of graft here, success or failure of the grafting or budding, that is depending on the kind of plant. So some plant is having the ability to form the successful union easily, and some plants did not have the ability to form the successful union quickly, difficult to form. So that case, the plant or the kind of plant and type of graft that can influence the success of the grafting. So some plants like oats or beeches, that is the temperate plants, so that is forming the successful graft union very difficult. But once the successfully it is formed, then it can grow simply without any problem. The forming is the problem. Then grafting and budding technique, that is influencing in the healing of the graft unit. So that is also very important. Especially we can go in budding, we will see patch budding and tea budding possible. It is highly successful in thick bark species in patch budding, and tea budding is very success in the species that is having the higher cambial activity. So some errors of the technical fault also influencing the success. If we wrap the grafted portion or budded portion delayed, so that can influence the success. Then while wrapping, if the wrapping is made as loose, so that is also not formed the successful. Even uneven cuts, because while incorporating the cyan into the rootstock, we have to make the even cuts, and the size of the cut should be in a position to join. So if we make the large or uneven cut, it can cause the problem. Even if the cyan, if we didn't preserve that cyan properly, some moisture loss occur in the cyan, that also influencing. So while doing, it should be close contact with the rootstock and cyan. So that is the case. That case we are saying the plant species or the type of graft, patch budding or tea budding, that can vary. Then we have the second factor, environmental factor, temperature. So that is very important for the callus formation. If the callus forms successfully, it will form the successful union quickly. So for the callus formation, temperature plays a major role. If the temperature is 32 degrees centigrade and higher, the callus formation will be affected. So that can, the higher temperature influence the callus formation. Due to that, the death of the cells also occur if the temperature is higher. So the appropriate temperature should be there while doing the grafting to promote the successful callus, and it will influence. Then if you look into the, especially in apple, grapes and pears, the grafting, that is the simplest techniques we can utilize these type of fruit crops and obtain the success very quickly. In mango also, even in tropical, we can obtain the successful grafting year round. The temperature is suitable for callusing, so we can do the successful grafting throughout the year. But for the mango, if we look into the good temperature, that is 24 to 28 degrees centigrade. So because the callus formation is influenced by the temperature, that should be, we have to consider. Even if the temperature is low, so below the 20 degrees centigrade or the lower than that, it will affect the callus formation. Above the temperature and below the temperature also influence the callus formation. Then moisture, because moisture also very important, because in science moisture loss happen, desiccation happen, it born from the successful callus and the successful union. So the moisture and water relations in plants also very important. If the desiccation of the cells happen, it will affect the success of the graft union. Then oxygen supply. The optimum oxygen supply is very important for the cell division, because cell division increase, then only it will form the several cells and form the callus, then it can heal quickly. So the oxygen supply also very important for the rapid cell division. Due to that, the healing process will be promoted. So these are the environmental factors we have to consider for the temperature, moisture, and the oxygen supply. So other factors, I think it's time up. So other factors we can continue in the next class. So today we started with the what is grafting, what is scion, what is rootstock, what is intermediate stock, callus, and what is graft union, what are the requirements we need to obtain the successful grafting, like compatibility, then matching of tissues, pressure, seasons. These are the things we have to consider. Then what are the factors that can influence the success of grafting, the type of plant or graft types, then environmental condition, growth activity of the rootstock. There are several factors I have listed. So under that, the first one is the plant type. So some plants form the successful union quickly, some form the difficult. So that is depending on the plant type. Like some temperate plants like peaches and beech, so oats, that is forming the union very difficult. But some apple, pears, and the grapes that can easily form even in mango. Then under the environmental condition, the mainly oxygen supply we have to consider, temperature, because temperature is important for the callus formation. Oxygen supply is important for the rapid cell division, so that is promoting the callus formation. Then we have to consider about moisture, because some desiccation happen in the scion, so that won't give the successful union, because we have to avoid the desiccation or moisture loss from the cut portion. So after this, I will stop it today. Then from next lectures, I will continue the factors that are affecting the grafting. Then we can move to the body. Thank you.
Thank you.
Thank you.